Have you ever wondered how scientists, actually figure out the root cause of a complex disease?
It's a huge question.
Right, because we hear about these massive medical breakthroughs all the time. But the actual mechanical reality of how researchers sift through, billions of data points to find that one exact biological target, it can seem completely impenetrable.
Oh, it's a monumental task.
Yeah.
Let's be real. The human body is a totally chaotic environment.
Yeah.
And finding the origin of a disease is rarely as simple as just spotting a single broken gear. You're usually looking for a really subtle flaw in a staggeringly complex, interconnected system.
Which is wild to think about. So to really grasp the scale of what we're looking at today, let's build an analogy. Imagine you are trying to completely understand the economy of a massive metropolis. But you are only allowed to look at its road traffic.
Okay, I like this.
Now, obviously, observing the traffic is incredibly helpful. It tells you where people are going, where the bottlenecks are, when rush hour happens. But what about the city's power grid? Or the municipal water supply?
Right, you're missing the big picture.
Exactly. What about the zoning laws that determine where factories can even be built in the first place? If you are only looking at the cars on the road, you are entirely missing the underlying infrastructure that actually dictates how the city functions.
And applying that directly to biological data, looking at just one type of information like gene expression, which we call transcriptomics, is the equivalent of only looking at that road traffic.
So it's just one piece of the puzzle.
Exactly. It's a critical first layer. But to genuinely understand how a disease takes hold, we need the power grid, which in this case would be proteomics, the study of proteins.
Okay, proteomics is the power grid.
Right. We need the water supply, which is metabolomics. And we need those underlying zoning laws, the epigenomics, which actually dictate how and when specific genes get turned on or off. Integrating all of these different views into a single cohesive model is what the scientific community calls multi-omics.
Multi-omics. And that concept actually brings us to the core mission of today's deep dive. We are exploring a super comprehensive 2022 survey out of the University of Nevada, Reno.
Yeah, it's a really fantastic paper.
Authored by Zainab Maghsoudi, Han Goyen, Alariza Tavakoli, and Tin Nguyen. And what they did was review 32 different computational approaches developed over a 17-year period, from 2005 to 2022, all focused on this idea of integrative pathway analysis.
And this is so important because to appreciate the cutting edge, we really have to recognize the limitations of the early pathway analysis tools.
Right, the early stuff.
Yeah, the very first generation basically just took a list of significantly mutated genes and looked for basic patterns. It was, very one-dimensional. Then the subsequent generations got slightly more sophisticated.
How so?
Well, they started looking at all genes, not just the mutated ones. And eventually, they tried to map out physical interactions. But the fatal flaw was that they were almost exclusively looking at just one layer of data. Gene expression.
Just the road traffic.
Exactly, just the traffic.
Yeah.
But a disease doesn't care about the boundaries of our scientific disciplines, A disease is a cascade.
Of domino effect.
Yes. A mutation in a gene changes a protein, which alters a metabolic process, which ultimately causes the illness. To capture that real-world ripple effect, researchers realized they had to combine independent studies, so multi-cohort analysis, and radically different types of biological assays, which is the multi-omics piece.
But, the logistics of that sound like an absolute nightmare. Let's say you have genetic data coming out of a DNA sequencer in a lab in Boston, right? And protein data coming out of a mass spectrometer in a lab in Tokyo.
Very common scenario.
These machines don't measure the same things, and they certainly don't speak the same data language. So how do these algorithms actually make them communicate without losing the biological meaning?
That specific challenge dictates the entire pre-analysis phase.
Yeah.
You cannot simply dump these disparate files into an algorithm and expect a result.
It's not magic.
No, not at all. The workflow has to be completely ruthless about quality control and translation. So the first step is aggressive filtering. You have to strip out low-quality data points and entirely remove biological pathways that just don't have enough measured genes to be statistically relevant.
So you're just throwing data out right away.
You have to. One of the tools reviewed in the paper, CancerMay, is explicitly built to analyze dozens of cancer datasets simultaneously. During its initial prep phase, it actively filters out anything that isn't a known cancer-related gene.
Wow. Okay.
It forcefully reduces the noise before the analysis even begins.
Okay, so you aggressively clean the data, but you still have that translation issue between the Boston lab and the Tokyo lab.
Right, and that requires ID mapping. which is arguably the most crucial logistical step. Think of a biological pathway as a highly specialized construction crew, right? With multiple genes working together to build a cell structure.
Okay, I'm with you.
Over the decades, different scientific organizations built their own databases, like KEGG, Reactome, or Biopsych. These databases act like independent encyclopedias, and unfortunately, they often assign completely different identification codes to the exact same gene or compound.
Oh, I see. So the K-key database might call a specific gene, like builder1, while the Reactome database calls that exact same gene worker A.
That is exactly the reality of the field. So these integration tools must function as a Rosetta Stone. They either come engineered with a built-in translator that maps the incoming data IDs to a unified standard, or they force the user to upload a manual mapping file.
And if they don't?
If this translation is inaccurate, the integration fails immediately. You'd be trying to cross-reference data that the computer thinks represents two entirely different biological entities.
That's a disaster.
Right. So once the translation is secure, the software moves to data matching. This is where it aligns the vast expression matrices. So the final master dataset shares common patients or common genes across every single omics layer.
Got it. Okay, let me do a really quick recap for everyone listening just to make sure we're grounded. We've got the pre-analysis phase. First, you aggressively filter out the noise, clean it up.
Yep.
Second, you translate it using ID mapping, so the Boston data and the Tokyo data actually know they're talking about the same genes. Third, you match it all up. But the paper actually spends a significant amount of time detailing a fourth step called pathway augmentation.
Yes, this is fascinating.
They mention it specifically to solve what they call the one-to-many problem. How does that complicate the prep phase?
Well, biology is notoriously messy and highly efficient. The one-to-many problem stems from mechanisms like alternative RNA splicing.
Okay, break that down for me.
Essentially, a single gene sequence doesn't always produce just one specific protein. Depending on how the cell reads and splices that genetic code, one single gene can physically code for several entirely distinct proteins, each with a different function.
Wait, really? One gene, multiple different proteins?
Yes. So if your software algorithm just ruthlessly merges all the data down to a single gene ID, you are completely losing the nuance of which specific protein variant is actually malfunctioning and driving the disease.
Because the computer just says, oh, it's gene X, without caring that gene X is currently making protein version 3 instead of version 1.
Exactly. You lose the resolution of the data. And you have a similar issue with microRNAs. These are tiny molecules that act like dimmer switches for gene expression.
Dimmer switches. I like that.
Yeah. And a single microRNA might suppress 5 different genes, or 5 different microRNAs might all target one specific gene. If you force that complex web into a simple spreadsheet column, you destroy the biological reality.
So how do you fix it?
To solve this, specialized tools like Mirror Integrator perform pathway augmentation. Instead of collapsing the data to make the math easier, they actually expand the underlying map. They inject predicted and validated microRNA interactions directly into the pathway model before the analysis even starts.
Oh, wow. So for anyone working with this kind of data, the stakes of this pre-analysis phase are massive.
Huge.
If you skip the translation mapping, or if you ignore things like alternative splicing just to save, computing time, you aren't just getting a slightly less accurate result. You're literally going to hallucinate biological connections that do not exist while blinding yourself to the ones that do.
The output is only as trustworthy as the preparation. There's no getting around it.
Garbage in, garbage out.
Exactly. And once the data is rigorously prepared, the actual hunt for the disease pathway begins. The authors of the survey categorized the 32 tools they reviewed into 4 distinct evolutionary strategies. The first 2 strategies are older, and they rely fundamentally on calculating p-values.
Okay, so a p-value being the standard statistical metric that tells a researcher how likely it is that their result is a genuine biological effect, rather than just a random fluke in the data. Like, the lower the p-value, the higher the confidence.
It's a perfect summary.
The paper labels Category 1 as gene-level integration. Walk us through the mechanics of that.
Sure. The tools in this first category, which includes software like KappaView and 3-omics, attempt to solve the multi-omics puzzle by zooming in on individual genes first. Let's say you are tracking a specific gene across a DNA study, a protein study, and a metabolic study.
Okay, 3 separate studies.
Right. The tool calculates an independent p-value for that gene in each separate study. Then, It uses established statistical formulas like Fisher's or Stauffer's methods to mathematically fuse those separate p-values together into one ultimate summary p-value for that specific gene.
Oh, interesting.
Yeah, it does this for thousands of genes, ranks them by their new summary score, and then checks to see which biological pathways happen to contain the highest ranking genes.
Okay, but when I look at the mechanics of that, a massive vulnerability kind of stands out. To calculate a summary score for a specific gene across 3 different studies, you need data for that gene in all 3 studies.
You're hitting on the exact problem.
Right, so if the protein lab in Tokyo just didn't measure that specific gene because of, I don't know, the type of assay they used, what happens?
The algorithm drops the gene entirely.
Wait, really? It just throws it away?
It drops it entirely. That is the fatal flaw of gene-level integration. It demands absolute consistency across every dataset you feed it. If a specific gene acts as the master switch for a deadly cancer pathway, but it happens to be missing from the metabolomics data layer,
The tool just discards it.
you lose the most critical piece of the biological puzzle simply because of a missing data point in one column.
Which logically forces the scientific community to pivot. They needed a method that wasn't so easily broken by incomplete data. And that brings us to category 2, which they call pathway-level integration, featuring tools like Impala and Reacto-MedGSA. How do these fundamentally change the math?
They shift the order of operations. Instead of trying to combine the p-values of individual genes across different studies, these tools analyze each dataset completely independently from start to finish.
Oh, I see.
Yeah, so the transcriptomics data runs through its own analysis and concludes pathway A is highly significant. The proteomics data runs separately and concludes pathway A and pathway C are significant. The software then combines those final pathway-level verdicts.
Okay, the flexibility there is totally obvious. You could take a study measuring DNA methylation and a study measuring protein levels, 2 things that are biologically vastly different, and combine them seamlessly.
Exactly.
Because you aren't forcing the raw data to align, you're just comparing the final conclusions.
The flexibility is unmatched, which is why these tools are so widely used. But, and this is a big but, there's a severe mathematical trap hidden in this approach, and the survey authors highlight it very clearly.
What's the trap?
Most of the statistical tests these tools rely on operate under the mathematical assumption that the different omics layers are independent variables.
But wait, we know for a fact that they aren't independent. I mean, the DNA code literally dictates the creation of the RNA, which dictates the creation of the protein. They're a deeply dependent chain reaction. Does assuming they're independent variables skew the final results?
It absolutely compromises the statistical integrity. When you treat biologically dependent mechanisms as independent mathematical variables, the formulas can easily generate hyperinflated significance scores.
Oh, so it thinks something is a huge deal when it really isn't.
Precisely. Furthermore, these p-value combiners frequently ignore a metric called effect size. And effect size is the actual physical magnitude of the biological change.
How much things actually moved.
Right. If you have a massive data set, a microscopic, biologically meaningless fluctuation can mathematically generate an incredibly small, highly significant p-value.
Let's bring that back to the city analogy for a second. It would be like discovering that traffic on a specific highway increased by exactly 1 car every single day at 4:00 p.m. for an entire year. Statistically, that is a perfectly consistent, highly significant pattern with a tiny p-value. The math is flawless. But economically, 1 extra car means absolutely nothing to the city's infrastructure. It's totally irrelevant.
That is the perfect illustration of effect size versus statistical significance. If you only combine p-values without weighing the effect size, your algorithm will flag dozens of pathways as critical, when in reality they are just experiencing meaningless biological background noise.
So the math is tricking you.
Yes. The field realized that treating biology like a pure statistics problem was generating way too many false positives. They needed algorithms that actually understood the physical architecture of a cell.
Which introduces category 3, graph transformation. If the p-value methods are basically just counting the broken parts of a car engine to guess why it won't start, graph transformation is actually mapping out the engine's entire wiring diagram to see exactly how turning one specific gear physically moves another.
That's a great way to look at it. Graph transformation algorithms are a massive leap in complexity. Tools like Paradigm or MosClip, they don't just look at lists of genes. They model biological pathways as intricate, interconnected networks, often called factor graphs.
Factor graphs, what does that mean in practice?
Take Paradigm, for example. It evaluates a single biological node by simultaneously calculating the gene's copy number in the DNA, its expression state as RNA, the resulting physical protein level, and whether that protein is currently active or dormant.
Wow.
It attempts to mathematically simulate the actual flow of biological information.
So it's literally trying to recreate the living cell inside the computer.
Yes. And the paper mentions ML's-CLIP does something even more targeted for clinical use. ML's-CLIP generates highly connected subnetworks known as cliques. It doesn't just look for what is broken. It specifically uses a method called Cox regression.
Wait, Cox regression. I've heard of that in clinical trials, right?
Exactly. It's a survival analysis tool. ML's-CLIP uses it to calculate the relationship between those multi-omics subnetworks and actual patient survival times. It directly links the multi-omics data to the clinical outcome, predicting how a specific pathway disruption impacts mortality over time.
The explanatory power of that is incredible. If it finds a target, you don't just know that it's statistically significant, you can see the exact biological domino effect that caused it.
You see the whole picture.
But, I mean, computationally, rendering a dynamic model of thousands of interacting genes and proteins must be excruciatingly heavy.
It requires immense computing infrastructure. Graph transformation tools are exceptionally difficult to build, difficult to maintain, and incredibly time-consuming to run. You are basically trading mathematical simplicity for biological accuracy, and the cost is paid in computational sweat.
So if the statistics are too simple and the graph networks are too heavy, the technology sector's inevitable response is, naturally, to hand the problem over to artificial intelligence. Which leads us to category 4, machine learning-based approaches. Tools like GSOA and pathway PCA. How does AI tackle integrative pathway analysis?
Machine learning discards traditional statistical modeling almost entirely. Instead, these tools utilize advanced classifiers. Let's look at GSOA, which utilizes a support vector machine architecture.
And SVM.
Right. The fundamental mechanism works like this. The algorithm isolates the multi-omics data for just the genes contained within one specific biological pathway. It then attempts to sort the patient samples into healthy or disease categories using strictly that isolated data.
Oh, I see. So it's essentially testing the pathway. It's asking the AI, hey, can you accurately diagnose these patients by only looking at pathway X?
Exactly. If the AI model can perfectly classify the healthy and sick patients based solely on the data from pathway X, the algorithm concludes the pathway X must be deeply fundamentally linked to the disease.
That makes total sense.
And they score this predictive accuracy using a metric called area under the curve, or AUC. The higher the AUC, the more critical the pathway.
Okay, the predictive power of AI is undeniably state-of-the-art. But earlier, we talked about how essential it is to understand the actual wiring diagram of the cell. If the AI is just sorting data into buckets, isn't it completely ignoring how the genes and proteins actually interact with each other?
You've identified the classic black box problem of machine learning. These models are incredibly adept at finding hidden patterns across multi-omics layers, but they often treat the pathways as isolated bags of genes.
Bags of genes, that's a great term.
Yeah, they do not model the biological domino effect. Furthermore, an AI's output is highly volatile depending on the specific algorithm chosen and the parameters the researcher sets. It can tell you with incredibly high confidence which pathway is driving the disease, but it cannot explain the biological mechanism of how.
Which is terrifying in a way. Let's do another quick recap and map out where this leaves a modern researcher. We've got a 17-year evolution of 32 different tools. You have p-value combiners that are fast but mathematically risky. You have graph models that map the biology beautifully but require supercomputers. And you have machine learning models that predict flawlessly but hide their reasoning in a black box. How does a scientist actually evaluate and select the right tool for their lab? The survey authors didn't just categorize these tools, did they? They rigorously graded them.
They did. And the evaluation metrics were strictly practical. They assessed the tools based on whether they've been validated on high-quality real-world data, the software's stability against crashing, the quality of documentation, and overall user-friendliness.
So who passed the test?
12 of the methods scored above a 4.0 out of 5, with tools like Reactome GSA, Paintomics 3, and Active Pathways really leading the pack.
What separated the top-tier tools from the rest of the pack? What made a 4.0?
Honestly, the inclusion of web-based graphical user interfaces.
Yeah.
GUIs.
Oh, so just making it easy to look at.
Exactly. The most effective tools allow researchers to interactively click, zoom, and visually explore the pathway networks directly on their screen, rather than forcing them to parse through raw terminal code. Usability drastically increases adoption.
But even if a researcher selects a highly rated, user-friendly tool, the paper stresses that the entire field is still slamming into a massive structural bottleneck. And it brings us back to the encyclopedia problem we talked about earlier in the pre-analysis phase.
The overarching challenge is that human biological knowledge is deeply fractured. It is siloed across dozens of competing databases. The survey points to a project called Pathway Commons as the theoretical solution.
What's Pathway Commons?
It's a colossal, unified repository. It has successfully aggregated over 2.4 million biological interactions from 22 different databases, all normalized under a single identification system.
Wait, that sounds like the exact Rosetta Stone the entire multi-omics field desperately needs.
It is the ultimate map. But the survey reveals a staggering reality. At the time of publication, practically none of the multi-omics tools fully and efficiently integrate Pathway Commons.
Really? Why not?
The unified data exists, but the computational software simply hasn't evolved to process that massive integrated architecture yet. It's just too big.
It's like having access to a hyper-accurate, real-time satellite map of the entire global economy, but your GPS software refuses to load anything bigger than a single city block.
That's a perfect way to put it. It highlights the huge gap between biological knowledge and computational capability. And dealing with that gap actually forces researchers to confront a broader philosophical shift in how they view data.
Oh, so?
Well, there is a deeply ingrained assumption in modern science that more data inherently leads to better results.
Right, which we could call the paradox of multi-omics. If we can measure the RNA, the proteins, the metabolites, and the epigenetics all at once, why wouldn't we just feed all of it into the algorithm every single time?
Because biology is deeply context-dependent. The authors highlight a pivotal survey by Kanzler and colleagues that tested this exact assumption. They ran 2 distinct case studies.
Okay, tell me about the first one.
In the first study, analyzing mitochondrial response, integrating 3 layers of omics data, was vastly superior. The multi-omics approach revealed critical metabolic pathways that analyzing just the gene expression completely missed.
So in that specific scenario, more data fundamentally solved the mystery.
Exactly. However, in their second case study, they analyzed murine hepatocytes, which are basically mouse liver cells. In that scenario, running the analysis on just the transcriptome, a single layer of data produced more accurate, biologically relevant results than the multi-omics integration.
Wait, the simpler, older method actually outperformed the fancy multi-omics integration. Why?
Because if a specific layer of biological data, say, the metabolic fluctuations, is not fundamentally driving the condition you're studying, forcing that data into the algorithm doesn't provide insight. It injects mathematical noise.
It dilutes the genuine signal.
Precisely. The algorithm ends up spending all its computing power trying to find patterns in completely irrelevant data.
Going back to our city, if you are strictly trying to fix a traffic jam on the highway, pulling the data for the city's water pressure and zoning laws isn't going to help you at all. It's just going to confuse the model.
Right.
Multi-omics integration is an incredibly powerful tool, but it is not a magic bullet that replaces critical thinking. You have to deeply understand the biological context of the disease before you decide which data layers to even unleash the algorithms on.
Context dictates the computational strategy. It always has. But as the tools refine their ability to filter noise and process larger unified databases like Pathway Commons, the speed at which we identify complex disease targets will just accelerate exponentially.
We've tracked an incredible evolutionary journey today. We started with the sheer logistical nightmare of forcing disparate lab data to speak the same language through ID mapping and pathway augmentation. We watched the field evolve from simple, flawed p-value combiners that ignored biological reality into massive graph transformations that literally try to digitally recreate the cell.
And finally into AI classifiers.
Right, AI classifiers that can perfectly diagnose a pathway without needing to understand it. Multi-omics integration is the ultimate architectural blueprint for discovering why diseases happen, but it demands relentless data prep and a hyper-specific matching of the software tool to the biological question.
It's an art as much as a science.
Which leaves you, the listener, with a final provocative thought to consider. We spent time unpacking the black box nature of Category 4 machine learning tools. As these AI models ingest exponentially larger multi-omics datasets, their predictive accuracy will inevitably approach perfection.
Undoubtedly.
We are rapidly approaching a horizon where an AI can look at your specific multi-omics data and tell us exactly which pathway is malfunctioning. But The mathematical complexity of how the AI found that pattern will be so deeply layered that no human mind will be able to comprehend the underlying biology. The AI will see the wiring diagram flawlessly, but we won't.
The ultimate black box.
Exactly. If a black box algorithm can perfectly prescribe a personalized cure by crunching millions of data points, does it actually matter if humanity no longer understands the biology behind it? Food for thought for your next deep dive. Thank you for joining us on this rigorous exploration of the sources. Keep questioning the data, keep challenging the algorithms, and we will see you next time.